So our last external speaker is Wendy Chung. Wendy is MD and Ph.D. She's a clinical and molecular geneticist and the chief of the Department of Pediatrics at Boston Children's Hospital and the Mary Ellen Avery Professor of Pediatrics at Harvard Medical School. Dr. Chung directs NIH-funded research program in human genetics of pulmonary hypertension, breast cancer, obesity, diabetes, autism, birth defect including congenital diaphragmatic hernia and congenital heart disease. She has led studies to improve newborn screening for spinal muscular atrophy, Duchenne muscular dystrophy, and now Guardian, the Genomic Uniform screening against rare disease in all newborn. She's a national leader in the ethical, legal, and social implication of genomics. She was the recipient of the Rare Impact Award for the National Organization of Rare Disorder, and she is a member of the National Academy of Medicine and the American Academy of Physicians. Dr. Chung received a BA in biochemistry from Cornell University, a MD from Cornell University Medical College, and a Ph.D. from the Rockefeller University in genetics.
Thanks so much for having me here today. Our first two speakers have done such a great job with the introduction that I'm going to zoom through some of the background, but I'm going to continue the story of undiagnosed individuals and how we can use some of these new technologies to get to answers for them. As you'll see, we don't have all the answers, but I do think we're making significant progress. So my conflicts here. You heard already in terms of thinking about some of the differences, similarities and differences, limitations of short read genome sequencing, especially compared to what we can get increasingly with long reads. You've already heard in many cases some of the advantages and disadvantages or differences, and I'll try and show you some of the differences between that. As we're doing this, I do think we're still not at 100% in terms of diagnosing those who sniff or who smell like they should have a diagnosis, but I think we're getting better. And we're starting to realize some of those regions of the genome that have been difficult to interrogate, some of which I have to admit, I myself didn't even realize I was missing as we were going forward. And so I think as we do this, we're going to identify even whole new classes of genes. And especially, I'll just say one take-home point for me is that this methylation signature that you've been seeing about, I don't think we fully tapped in terms of being able to use that as a direction to point to where some of these answers are hiding. So within this, hopefully it's already obvious, but as we do the genome assemblies from short reads, it suffers from some limitations. If you have repetitive regions in particular, if you have regions where you've got duplications of the genome, there can be things hiding because your alignments really just aren't showing you what's there. You may have things that are essentially going in the garbage when you're doing the alignments, and so you're not recognizing what's there. Or if you have regions where you have very similar sequences, you may have such low read depth with the variant that you're seeing, it essentially looks invisible to you. So with this, you've got a lot of ability now because of the length of the reads to uniquely map these in the genome and again be able to see things that we were missing before. You've already heard a lot about the epigenetic marks that we're seeing, some of the structural variants, as well as haplotyping or phasing. I'll say that in particular through some of the things that we had done through newborn screening, I wish I'd had more of that haplotyping and phasing to be able to see that for some of the recessive disorders. So I won't go through it again, but I will say this is, to me, was some of the most creative chemistry and technology when it first came out. I will timestamp it. It's been around a while. It's taken some time in terms of being able to develop, especially the analytical tools for this, but I do think we're on a trajectory being able to use this information more, and it really is just incredibly elegant chemistry to not have to do any amplification and to literally read the intact strand and to see those chemical changes as it's going through the pore and read out, therefore, essentially chemical modifications to the nucleic acid that you're reading. Just being able to do this in real time. For some of you, we haven't talked about it today, but there are even technologies to be in real time reading what you're seeing through the core, kick it out of the pore if you're not interested in reading it, and therefore being able to focus and interrogate your sequencing reads on one part of the genome that are really quite elegant. So as we're doing this and thinking about how to do this, there is the ability to flex in terms of doing relatively small numbers of samples, doing them relatively quickly, being able to do larger data sets and doing them with greater comparisons. I do think, as was stated earlier, the more that we have comparator data sets to be able to compare against, the more we will be able to recognize what's different about some of our cases. And so there are some of those large genome data sets now coming online, not just 1,000 genomes, but all of us and other data sets that are going to help in terms of that. I'm not going to go through in detail. You've already been hearing in terms of the pipelines to put your data through in terms of this. I will say these are in rapid evolution, and in a good way, I think are continuing to improve, as I said, especially as we think about some of the structural variants and some of those epigenetic marks. Within this, let me just point back. What I'm going to tell you about today is from Boston Children's Hospital, a cohort that's been, I'm a relative newcomer to the community, there for about 1.5 years, and I won't take any credit for it. But one of the wonderful things about the community is really the spirit of working together. And so one of the things we've created is the Children's Rare Disease Cohort, which takes then cohorts, several different cohorts collectively, but putting them in a common data pool where we can collectively work and help each other in terms of being able to diagnose those undiagnosed going forward. And that's proven to be extremely effective, and I'll show you about this today. One of the nice things that we've done at Children's, just in case it's helpful for any of you, is to have a common platform. So we fund it centrally in terms of having a common platform for any clinician to be able to use the tools that we build centrally. That way you don't have to be a genomicist in your own laboratory. We're centrally supporting that, constantly updating it as we're adding new tools, new ways of sequencing. Again, being able to democratize this for our clinicians, being able to take those individuals who know how to recognize something in the sequence they're seeing, but not necessarily do the huge informatics lift behind. All of this in essentially a browser that's very intuitive, hopefully, and able to see in terms of being able to browse this. And as I said, making it much easier, bringing more tools to more people is sort of the thinking in this. As we've done this, we did annotations that are similar to what many of you are used to looking at, and in doing this, trying to automate this system increasingly so that even as we're individually analyzing a case, we're surfacing the rare variants that are most likely to be phenotypic matches by pulling the current information from the EHR with the HPO terms and being able to use AI to then surface and prioritize those variants. So again, trying to think about doing this at scale and actually now in a systematic way going back and rechurning our data sets on a approximately annual basis. So what I'm going to tell you about today is a portion of the CRDC where we decided we were going to try and diagnose the undiagnosed. This was many of our investigators who came together. Shout out to many of them. I'll just specifically point out Alan Beggs, who's been working on muscular dystrophy for decades. Alan has a unique cohort of individuals with true muscular dystrophy. He's done an incredible job in terms of phenotyping them. So the prior probability for essentially that entire cohort extremely high that there's going to be something genetic. I'll take the blame on the other side. I look at many congenital anomalies, and in many of those cases, I'll be honest, we don't really know the underlying genomic basis for those conditions. They may or may not be due to single gene conditions, and so you'll see some of the differences in terms of the diagnostic yield, but I put in some of our cohorts in terms of diaphragmatic hernia, esophageal atresia, and then we have many, of course, as many of you do, individuals with neurodevelopmental issues or neurological symptoms, and then just a potpourri of various different undiagnosed conditions. As you'll see, and this is true for many of us in terms of read depth, about 30x is sufficient in terms of the depth of coverage you need to be able to cover this, and the strand length approximately 15 kilobases. Many of these samples over a decade old. So just in terms of those of you who may have samples in your freezer to be able to go back to, still be able to dig back into those old samples and come up with some answers. So this is overall being able to see what happened with the cohort. I'll go in cohort by cohort, and I'll show you some individual cases at the end. Still to say the majority, in other words, more than 50%, we still don't have a diagnosis. So just to level set in terms of that. On the other hand, it's pretty incredible how many diagnoses we did find reciprocally. So this is not just a onesie and twosie. There's really a large minority of individuals that we now have diagnoses, and they really do fit. As we're going through I'll show you these in many different ways of slicing and dicing the data, but I do feel like what I'm showing you is the minimal in terms of the minimal yield. We were pretty conservative with the interpretations. So starting out, for instance, this is congenital myopathies and muscular dystrophies. This is, again, Alan Begg's cohort. This is the best of the yield, just to set expectations. Again, the prior in this case extremely high. Still, individuals who are unsolved at the top, but you can see a number of different, and I'll go back to this again, individuals due to large deletions, unmappable structural regions, areas where we were able to get phasing resolved. There might have been one mutation, as you'd heard before, and identifying both the second and the phasing, repeat expansions, and then complex structural rearrangements. And for any of you that are interested, these specific genes involved here, and I'll go back to a couple of those cases. As I was alluding to before, for diaphragmatic hernia, the yield not as high, likely because the complexity in terms of the genetic architecture more complex. Some of these due to single nucleotide variants, others due to more complex structural rearrangements or complex indels. Esophageal atresia, again, within this, still the majority not yet diagnosed. Again, the underlying complexity for many congenital anomalies, probably more complicated. Certainly CHD7, a well-known gene for many of you in terms of esophageal atresia, hit recurrently, as well as some complex structural variants. For those with neurological disorders, again, the majority still not solved, but perhaps not surprisingly, a repeat expansion as well as single nucleotide variants. And then I'll highlight one of these cases, but again, structural variants that we're seeing as well as duplications. So overall, in terms of the cohort, for those of you who want to be keeping track in terms of what you're seeing, still, again, we see a variety of different types of conditions, different types of variants, that perhaps weren't seen on short read sequencing. As we go through and look at this and look at the yields by the different cohorts, this is what I meant by that it's still the minority, but overall it's now a quarter of the cases, 24% of the cases where we didn't have a diagnosis before, that with this technology we're able to see and call things that we weren't able to before. So not bad for me in terms of, I don't know about you, but the families that are coming back to me year after year in terms of what are you going to do for me lately? These are the things that we're trying now in terms of solving those cases. So for the ones that we're trying to think of in terms of where is long read really uncovering and giving us additional value, some of these cases are highlighted here. And again, as we think about this, they're cases that have the methylation patterns are giving us, I would argue, an orthogonal way of reading this out. It's essentially a functional view of the genome in addition to what we might see from some of the variants. Makes me increasingly confident that what we're seeing is really causative, as well as I'll get to in a little bit, some of the more complex structural variants that we're seeing. These are the ones that I think the long read technology, there's no way. In going back and looking at our short read sequencing, standing on my head, doing other things, there's no way in the world I would have picked this up from what we did before. And those are things where we didn't have parents to be able to get the phasing resolved. We had issues in terms of especially the repeats, some of these complex regions of the genome, and I'll show you one of the case where you've literally got duplications and you can't uniquely map them back to be able to see. These were things that we could not have been able to see based on our previous data that we had. Within this, I will also admit that some of these things we did see when we went back to some of our old short read data, but for all of us, it's amazing what a tincture of time will do. That is that the same, even if it were as little as 2 years ago, the same variants that I saw with new ways of being able, new genes that have been identified, and I will say personally for things like alpha missense in terms of being able to prioritize different variants, especially missense variants, I think we're just able to recognize much better. So part of this is admittedly the technology. There's another part of it, which is our knowledge base and our general ability to interpret variants over time. So a combination of the two of those were incredibly powerful. So let me relatively quickly go through 3 cases just as examples. This was a little girl who came to see me when she was a baby with overgrowth, and in doing so, really didn't have particularly distinguishing features. I thought she might have Beckwith-Wiedemann syndrome when I first saw her. We had done the usual BWS, and you've seen some of those assays already, and didn't come up with an answer, but she still bugged me that there was something that was going on. We went through and did her sequencing here, and I'm sorry, my pointer's not going to work very well here, but she's on the top, and you can see the read depth coming down in the middle there, and that's the region where she has a deletion. Her mother and her father on the bottom. You can see that they have, this is a de novo deletion within that region. They have normal read depth. This particular region was in the 3' untranslated region, so whether or not it was actually functional was not clear to me. So this is where I bring in the methylation pattern as being a good readout. As you've seen before, the red being areas that are hypermethylated, the blue areas that are unmethylated or less methylated. And again, I wish my pointer were working here. In the middle here, you can see what's called haplotype-specific hypermethylation. And what you'll see is that for her reads, you can see more red than blue in terms of being able to see the regions that are hypermethylated. And in that particular case, hypermethylation actually increases expression of this gene. So she has overexpression of this gene. Why does that matter? Well, there's a condition for this particular transcription factor. It's interesting that there's a sort of Goldilocks in terms of being too hot or too cold. So it can be reciprocally associated with something that looks like Silver-Russell syndrome in terms of lower growth, or on the other end, Beckwith-Wiedemann syndrome in terms of on the higher side of growth. And in particular, although I'd seen her as a baby as I got these results, called up her mom to see how she'd been doing, and she said, you know, the funniest thing is her teeth. When she was a baby and saw you, she never had this tooth issue, except now she has so many extra teeth that her dentists don't know what to do. And in fact, that was the clincher for me, because that is a very, very specific finding that's associated with this particular gain of function or overgrowth for this gene. So that's just one example of what this can do, but again, something that we hadn't seen before and hard to know what to do sometimes for these regions that may be in regulatory areas. This was one of Alan's cases that was an 18-year-old, which on muscle biopsy clearly had muscular dystrophy. In this particular case, he knew one mutation from short read sequencing, trying to figure out whether or not there was a second mutation that might have been missed. And so you can see on the right over here, the region where there's a little bit of a gap. You can see a deletion of a few nucleotides, and then on the left in the areas of blue, a complex duplication. Now being able to put these together and see whether why we didn't see this before, and I have to admit this was one of these new things to me. This gene has a region where it's several exons that are entirely duplicated. So it's not just one exon, but it's a chunk of exons that are duplicated over 3 times within the gene. And so if you have a mutation in one of those exons, as you pile those together with short read sequencing, it basically gets lost in the noise. You don't see and you don't uniquely map. On the other hand, as you have the long read sequencing for this, you can actually see each of the strands individually, and you can read them out, and you can clearly see these regions. So there are regions like this I'm sure that many of us are just not used to looking at. If you now put these two together in terms of being able to understand the haplotype, you can clearly see the 2 different types of mutation, and you can clearly clinch the diagnosis in this particular case. Finally, I'll just say that this is one other little girl with a myopathic process that wasn't clearly defined. This is, again, a region where this particular gene, Notch2-NLC, can be associated with a repeat expansion. She is shown in the middle here. The regions that are purplish on the left there are the repeats, and the number of those repeats are what are shown in the box. You can actually see, like an accordion, that each of her alleles has different numbers of repeats, so there's also probably some somatic instability with this. None of those repeats on her father's side on the bottom, and a smaller number of repeats for her mother. And then you can see, again, this beautiful functional readout in terms of looking at the methylation pattern with that red showing, again, that she's hypermethylated in the region, unlike her mother, and probably the reason why she appears to have something that is not seen in the rest of her family. So just to briefly wrap up, in doing this, it is, not perfect. No technology is, but it's starting to show us and help us recognize things that we haven't seen before. My prediction is that we're increasingly going to use this. You've seen how it's being used on the UDN or used in more in a research phase, but I won't be surprised as some of you, possibly librarians, are looking at this, that you may start incorporating this more in terms of diagnostics as we go forward. Most importantly, just to thank all of the incredible people who've been part of this. I just get to be up here and be able to let you know about it. So thank you.